So here's my disclosure statement. I basically have no conflicts or any potential bias, either real or perceived, in this particular topic area today. So I'll start from the standpoint of the paradigm of risk, and we've heard a lot about this morning. It needs not much more introduction, except in the concept of what I do, we are interested in designing assays to assess toxicity from the standpoint of pharmaceutical materials, but we've learned a lot about that and how nanotechnology can, in fact, work with non-pharmaceutical materials. And I think from that perspective, it's useful to understand that in our assay design, we use the same kind of exposure hazard identity, but the problem is that articulated this morning, is that the exposure is relatively unknown, and therefore the idea of harm and what constitutes harm can be validated, but the exposure and therefore the risk of producing that harm is very, very difficult from the standpoint of understanding the exposure to nanomaterials. So in this concept, it's not unusual that we would go through understanding what the particles are that are presented to the oral cavity, what their presence is, what their uptake capacity is, what their transport and fate and toxic effects, and many of the things that we discussed this morning are in fact very familiar. And the idea in the pharmaceutical industry is that we could produce dose relationships where that exposure is known. But when it comes to oral ingestion of food, that's a bit outside my realm, however, we don't really know what the exposure is, and I'll try to produce that later. So in terms of defining harm, in the Code of Federal Regulations, safe or safety connotes a reasonable certainty in the minds of competent scientists. I think that's an oxymoron sometimes, but the substance is not harmful under the intended conditions of use. So one familiar example in my field is that of non-preventable or considered non-preventable medical harm is a medication side effect. So assuming that a drug is given for an appropriate indication, like an antibiotic for a rash, for example, and that'd be classified then as harm if there was a side effect. However, the risk of infection persistence from that rash and the morbidity of that rash weighed against the risk of doing harm, that is, experiencing that side effect, in this case favors administering the drug. So oftentimes in pharmaceutical industry, we're faced with not giving the drug versus giving the drug, and knowing the dose response and risk relationships, we can understand then the intentions of doing well versus doing harm. So in distinguishing then preventable harm instead of all harm as a target, which is my preference, then the former is actionable. That is, preventable harm is actionable, whereas preventing all harm is almost impossible. So that the resulting number of cases of preventable harm is then large enough to be considerably compelling to address, but it's not overwhelming like it would be in the case of all harm that would be paralyzing to producing any type of action forward. All right, so now I go back to Gut 101, and we talk about, in the case of oral nanotechnology, we have to understand a little bit about how this works. So in our intestines then, we've got lots of different cell types, and the epithelia then comprise lots of different things that I'll talk about, including most of the cell types that encounter our foodstuff, which are enterocytes. Now what's important here is that I'll emphasize continually this very small minority of cells called the M cells. The M cells are basically an immune interfacial feature that uses to process solid particles, like bacteria notably, and present them directly then to the underlying phagocytic cells or immune surveillance system that underlie our intestinal membranes. So in large part, if I could just make this black and white for you, the enterocytes that comprise 99% of our cellular lining of our gut really have very little efficiency in processing particulate matter. Particulate matter is basically the job of the M cells, but they only represent 1% of the cell population there. The rest of the systems can absorb soluble things, but the M cells are designated to absorb the insoluble things, and many nanomaterials are not soluble, so they're processed by that cell population. The other thing to appreciate is the fact that these cells that I just talked about are covered with goo. And that goo is a natural protective layer called mucus, and the mucus is in fact very highly heterogeneous. And it's not only heterogeneous from front of our gut to back of our gut, but is in fact heterogeneous in youth versus elderly, and is certainly heterogeneous in people who have pathologies like, for example, inflammatory bowel disease. So there's a lot of difference in heterogeneity in the patient population in how they present mucins. And how our food then is digested is that it must cross this mucin barrier to present itself to cells. The soluble fractions of food then can be absorbed by the enterocytes. The insoluble fractions are processed by the M cells. All right, so here's the last part of Gut 101, is that the mucin present in the stomach is very different than the mucin present in our upper GI, that is just the exit to the stomach, and certainly very different than the colonic mucin. So we have different types of barriers to help us process food and nutrients, and this is precisely the same barrier that nanotechnology must negotiate. Remember, it's no different in how it must cross into our system. So the other thing then is we look at the M cells a little bit differently now. The M cells are juxtaposed to our immune surveillance system in a very important way. Because they process things intentionally like viruses and microbes, they are positioned against then our dendritic cells, our B cells, and our T cells, which represent our immune surveillance system. So there's a direct handoff of this particular packaging mechanism through the gut wall to the cells that are there to process it and determine friend versus foe. And from friend versus foe, then, these signals are processed, go into blood circulation, upregulate or downregulate appropriate immune signals, and send them back to the gut to tell friend versus foe. So there's a circular communication that goes on via these M cells in the gut, but remember, they're only a very, very small fraction, 1% of the entire intestinal wall. So how do we do this in a laboratory situation? I just discussed how we have this very complex set of intestinal epithelia, the unstirred mucin gelatinous layer that's a barrier, the lumen where the food flows by, and underneath then is where the stuff has to go, the capillary blood flow that then takes all of those nutrients and takes it to the biliary processing system in the liver for the first pass effect. But in the lab, in fact, we've simplified this to look similar, but in fact, functionally, it has some deficits. So we take these epithelial cells and we seed them onto a porous plastic layer. That porous plastic layer is very stiff, but it has holes in it, and there's an unstirred water layer underneath, so that when we address the outer portion, the apical side, as we would in the intestine with soluble substances, they can encounter the epithelial cells from the intestinal source here in a cultured in vitro way. But the passage of this material into this layer is different, and the environment in which it's processed is different, and there are many subtleties to this system that make it non-equitable in terms of absolutely determining how things are bioavailable in our stomachs right now versus what they are in a Petri dish. So here's just one example. We have jejunum from the actual intestine compared to CACO2 cells, which are a colonic cell line. That is, it's immortalized. It's been immortalized to reproduce very, very quickly. And notably, it contains no mucin production. So if you remember 3 slides back, the mucin in the colon is in fact the thickest mucus possible. But these cells, these CACO2 cell monolayers, have no mucin production. Yet they are the mainstay of the intestinal absorption industry in terms of assessing toxicity. So if we look at CACO2 cells and look at their production, or so the processing of a common calcium ion channel blocker is just one example. You can see that if you compare the bars to the actual intestine, the bar shapes look very, very similar. That is, the apical to basal transport of the drug and so forth. I don't have time to go into the details. Looks very similar until you look at the y-axis. And you look at this y-axis tells us the amount of permeability, and now we have then a 10-fold difference in how the intestine passes that drug through it, as opposed to this intestinal cell monolayer that's supposed to mimic how this works. So while form looks similar, in fact, the absolute magnitudes are orders of magnitude different. All right, so now we bring in nanotechnology. Nanotechnology has found, and no pun intended here, sound resonance with the political and business sectors. This is a Cornell nanotechnology nano guitar. It's the size of a single cell, this whole thing is, and these strings actually move and can be played with a laser if you wanted to. So this is the nano guitar, and of course, this is the extreme of what nanotechnology is doing with taxpayer dollars that makes, the taxpayers very, very happy about this kind of thing. But the point is that there's an enormous diversity to the type of nanotechnology that might be encountered, that might be fabricated in the lab, and we refer to this as engineered nanomaterials. And the distinction is very important because what I'll show you later is that there are endogenous foodstuffs that have lots of nanoparticles in them naturally, and yet now when we introduce engineered nanotechnology into food, or have this idea of that's what we want to do, that there's an enormous new diversity of materials that the body ostensibly has never seen before that have to come and be considered for safety and toxicity profiling. So this is just one example. For example, you could think about making a new formulation of Mountain Dew with this nanoparticle that glows in the dark. No, you don't want to do that. So anyway, the idea is, so what is oral nanotechnology good for right now? What's it doing in the clinical arena right now? Well, so we take this problem, which is a classic pharmaceutical industry problem. 70% of all new drug candidates discovered by my chemical colleagues don't dissolve in water. They're called brick dust. That's the euphemism for new drugs these days, is brick dust. 40% then of those that then look promising in cell culture assays to fight cancer, to fight epilepsy, to fight heart disease, most of these also have solubility problems. So if they don't dissolve in body juices, how can we make formulations that can sell and be efficacious to patients? So nano-sized particles, however, have been found to dissolve very much faster than their larger particle cohorts, because they present a larger surface area, because they have a larger contact area with the juices of our intestine, and because then they can rapidly dissolve. So here then shows what we call a dispersion diagram that says as we move from the micro scale to the nanoscale, particularly now in the 100 nanometer regime, we see this exponential increase in the aggregate surface area per gram of nanomaterial. And we take advantage of this exponential part, particularly in the area between 100 and 10 nanometers, where we can get enormous size. We can get soccer field size per gram surface area from these particles. And how does that help us? Well, if we think about the intestine as a tube, now we have large particles, and as they transit the intestine in, say, 6 to 8 hours, they have a 6 to 8 hour chance then of dissolving and being absorbed through the intestine into our circulation. However, if they have a very slow dissolution, or they don't want to go into water, most of this material exits unchanged. Yet if we dissolve it or prefabricate it now into nanoparticulate form with enhanced surface area, we get far more absorption through the intestine in very, very small forms than we do in the big form, and that allows us then to produce increased formulations of drugs that normally don't dissolve in body fluids. So there's a number of FDA-approved drugs that, for this point of this talk, is only to show you that this is where most nanotechnology right now is involved in improving the dissolution of substances that don't dissolve. And I think this is useful to you because there are many micronutrient problems and there are many flavor enhancement problems, maybe other things that you can forecast, where difficult to dissolve substances may be useful in food, could in fact benefit from this type of technology. So we improve then dissolution in the gut for poorly soluble substances, and this improves our agent absorption through the gut. And this is relevant then to applications where dissolution, that is the rate of dissolving, is rate limiting to the GI uptake. So the drug readily permeates into the intestine and into the portal vein. Once it's dissolved, the involvement of the particle after you do that delivery is irrelevant. So if it is an insoluble substance that's simply carrying another substance, then right now the particle, for example, an adjuvant in a vaccine, will, as aluminum oxide, will deliver the stimulatory molecules to create the vaccine response and then get flushed through the GI tract and emerge largely inert. So aluminum oxide as a carrier is very interesting. It carries the vaccine through, it delivers the stuff to the epithelial wall, and then it just flushes through. So in that case, it's not important what happens afterwards. The particle excreted as an inert thing. Micronutrient dissolution strategies would follow the same concept, and natural product nanocarriers, such as solid lipid nanoparticles, SLNs, increasing population of these species coming about, as well as nanostarch type of particles being used to capture flavors, capture catalysts, capture enzymes for enzymes efficient people, and so forth. All right, so nanoparticles dissolve in the gut to improve the absorption. But the evidence for direct oral absorption of particles, that is insoluble stuff, is generally unimpressive. And for therapeutic purposes, it's really irrelevant. Therapeutically, we can't get stuff at a therapeutically important level through the gut. Why is that? So there are two basic issues. One is we can't get enough stuff across the intestinal epithelium. It can't go through the mucin very effectively. It can't pass the intestinal wall very effectively. There's a poor translation also of all of our in vitro data, which we heard about this morning. This connect between what we do in the lab and what actually happens in the human patient cohort is so miserably poor that we really don't have a lot of predictive models for this. And the influencing factors for nanotechnology. size matters, right? Surface charge matters. Whether it's positive particle or negative particle makes a difference. In Boulder, where I used to be an instructor in Boulder, we had a different discussion about positive, negatively charged particles. Not because of the marijuana laws, but there's another story I'm not going to get into. All right, hydrophobicity and hydrophilicity, that is the wettability with water or not, make a difference. The stability of how these particles interact with protein goo and with all of the goo in the stomach, like the mucins. And then the model validation, the KCO2 cells. I showed you that in many cases they look good, but in fact they don't replicate what we see in human beings. So I go back then to the model, and we're going to focus then because insoluble particulates are processed by this component of cells there. So what do we know then? In the intestine, let me go back. Here's the villus, plural villi there. So in the villus tips, we can get some particle size uptake because there's many enterocytes there. But as I told you before, they're very poorly efficient at processing particles. Their uptake of those particles is slow. The intestinal macrophages that prowl our intestines for germs and bugs, they're capable of taking up particles, but it's very slow. The enterocytes, as I said, are very slow. And then the Peyer's patches, where the M cells exist, it's efficient, but the cells are very, very small amount. So the factors, again, affecting that particle uptake is what I told you about: particle size, surface charge, and then there's a whole other area in which we talk about the animal model. Is the animal fed or fasted? That is an enormous difference for us in the pharmaceutical industry in terms of the uptake of particles if the animal is fed or fasted. The age of the animals, the exuberant young animal will digest particles much more effectively than the elderly animal. The species used we talked about this morning, the rats and the fact that the mice don't have gallbladders and so forth, this is an enormous difference for us. So generally in the GI tract, the uptake of particles is traditionally miserable. Less than 10% and often less than 1% of any solid particle dosing is orally bioavailable. That is transported as the particulate form through the gut and found in the bloodstream as a particulate form. So there's lots of different anecdotes about this. So we talked about the fact that the intestinal epithelium can be both friend and foe. It's a friend in protecting us. It's a foe in now not letting things through that now in our 21st century engineering wisdom we would like to push through. The mucus layer is thick and blocks then the invasion of those things as a friend. The enterocytes, they are very, very prolific. They migrate, they move, they are shed every few days. So even if the enterocytes are capable of taking up nanotechnology, the fact that they're shed every 2 days means that any particle burden that's been taken up by an enterocyte then gets sloughed and exits in the feces. And in fact, even though you got it into the enterocyte, it's now gone and not bioavailable. So I'm going to skip that. So the Peyer's patches are covered with then these M cells. And the M cells, as I've told you, are highly important, but they comprise a very small fraction. So here's a very beautiful study that I was able to review several years ago now from Germany, where they made a very exotic gold radioactive species, unusual because they produced it in a nuclear reactor. And they fed then gold nanoparticles made with this transient radioactive gold to rats. And they looked at the amount of translocated gold from outside, that is inside the gut, but then into the blood circulation, the percent translocated of total dose. And because of the sensitivity and radioactivity of this, they were able to do the first 100% accounting of where all of the gold particles go if you stuff them down the esophagus of a rat. So the idea is that size matters, that large particles small particles went more than large particles, and the point is that if we look at the total percent, I don't know if you can see in the back, but 0.5% is the maximum amount of dose that went anywhere. That means 99.5% went into the feces and didn't go anywhere else. And charge matters, as you can see. Negative charged particles were translocated far more than positively charged ones, but again, the y-axis is miserable, showing that in fact it's highly inefficient, even for particles that are 1.4 nanometers. That's just like 100 clusters. So we just completed the first in-human oral dosage study in our clinic, and we fed willing fraternity boys for beer money. We fed them silver nanoparticles under IRB approval in a consent form that was legally valid. And we took 60 and fed them oral dosing, and we did everything you could do. Serum counts of silver, blood counts of silver, MRIs, we did the whole shoot match, liver tox profiling, and for different dose regimens done, and we did a crossover study, so neither arm knew if they were drinking just pure water or drinking pure silver and water. And basically, the bottom line of this is that we found no silver detected in the urine, independent of the dose. We found some silver detected in the blood, but it was only ionic silver, not nanoparticulate silver, and we found no clinically important changes in any metabolic, hematologic, urinalysis measures, and we've got 3 papers published out there this year in this first-in-man study. So orally, we found nothing with silver. So now when we get to foods, all foods, either naturally or processed, contain nanoparticles and nanostructures. Milk contains caseins. It's a milk protein that aggregates. It produces then these complexes of milk and milk fats, and they make nanoscopic droplets. That's part of the production of ice cream and other milk products. Meat contains protein filaments that are insoluble, less than 100 nanometers thin. The organization and change of these collagenous and elastin-like structures affects their texture and the properties of milk and meat. And manufacturing also produces nanoparticles in foods. This includes key features of margarines in their leptic properties, how they touch the tongue, how we know it's Safeway ice milk versus $30 a kilogram Haagen-Dazs ice cream. It's all a leptic feature of how those fat droplets are processed. Toffees, chocolate, and cheese, all these things have then these nanostructures. But this is very compelling new data that I think is interesting. So this Indian group just published a paper cited here in which they found frequent nanoparticles of carbon in all carbohydrate-based foods that they cooked. Caramels, breads, jaggery, which is an Indian delicacy, sugar caramel, cornflakes, and biscuits. And you can see the nanoparticles here in black, 50 nanometers. Here's the particle size distribution. It's very, very impressive where they found no matter what cooked carbohydrate product they found these carbon nanoparticles in the form of 4 to 30 nanometers. And they say that then this is a ubiquitous thing that we have been living with since we have been cooking toffees and cakes and things like this, that we've been exposed to these particles ubiquitously over time and no one ever cared. And then the other notable is that the nano food market is expected to soar to an enormous market by 2015. You're in a much better position to call BS on these figures than me, but in fact, what I see when I survey out there, that packaging in the form of nano silver in terms of all of these food storage surfaces. There's now an entire Korea and Japanese appliance line that's entirely coated with nano-silver particulates in the refrigerator. There's lots of then nutraceutical supplements and expanding to ingredient functionality, emulsions, and even to sensors that sense freshness versus spoilage. So examples are the anti-inflammatory properties of curcumin. They're enhanced if it was emulsified droplets versus below 100 nanometers in size. So curcumin, as you know, as a nutraceutical. Nanosilver that we deal with in our lab is very popular. In fact, I would say it's the most popular from the standpoint of what I see coming to journals that I review. Nanoclays are now incorporated into plastic bottles for drinks that prevent oxygen migration, oxygen diffusion in plastic bottle walls that might destabilize the drink. So they want to extend the product's shelf life. Nanoencapsulation is used to improve the solubility properties and enhance bioavailability of flavors, for example, and other additives. And then there's this curious product from Israel, a canola oil derivative that's used, they claim to make nanocapsules and nanomicelles of the phytosterols in this oil, and they're thought to then reduce the uptake of cholesterol from the digestive system. Obviously not something claimed in the United States, but founded in Israel. So in our case then, we're not dealing with that, but we deal with what's called engineered nanomaterials, things that we actually make in the lab, like nano silver, something that doesn't exist in nature, where you have to do the chemistry, clean it up, ask what are the stabilizing agents, what are the additives, what are the contaminants. Then we do the analytical chemistry to understand where are those contaminants, what is the shape, what's the functionalization, is it positively charged, is it negatively charged. Then we do the biological and environmental context, and we try to do this and we extend it across the continuum of simple in vitro tests to predictive ex vivo tests to predictive in vivo tests in animals, and then do in vivo testing in humans. And that's the pharmaceutical paradigm, and I presume that your community works the same way. So in vitro methods then are used to assess this exposure and this risk and this toxicity, and we use mostly cell-based assays, where we look at cell adhesion, cell entry of the particulate, cell processing and trafficking of the particulate, the signal production, the stress signals that might come out of that cell, like cytokines or reactive oxygen species, like singlet oxygen, cell toxicity and the viability that the cells die, of course, and their phenotypic changes. Do we change the cell's behavior by feeding it particles? So what we have to worry about is the fact that the cell lives in a protein milieu that's about 4,000 different serum proteins in any given milieu in our body. And so these protein interactions with these materials are interesting because they condition the materials to enter the cell. So the cell never sees a naked particle. By the time the particle gets to the cell wall and is processed by cell receptors, it's coated with hundreds, if not thousands, of proteins on the surface. So this conditioning interaction is very important. So observation number one here is that nanomaterials interfere with these assays. Oftentimes, all of these assays that we've used for cell culturing for decades, now when we put different nanoparticles in, I don't have time to go through this, but the slides are available for you. I try to make these references available. There's lots of things that signals where the gold or the silver or the carbon nanotubes or whatever the material is, it actually interferes with the assay, so we get false signals from the traditional assays that we've always used by putting the materials in that complicate the assay. Observation number two is that nanomaterials change their physical state once we put them in biological milieu. So here's carbon nanotubes in water. Here's carbon nanotubes in cell culture milieu. In cell culture milieu, after several days, we can actually see the carbon nanotubes start to melt together and form these agglomerates. So what we thought we were presenting to the cells actually is nothing of the sort. And the entire size scale changes of these interactions, and the entire physical state of these things interact. And so you can think about how asbestos causes fibrotic lesions in the lungs, and the fact that the morphology of asbestos is so important to how it creates those lesions, and that all silicas of a different morphology don't create asbestos complications. So you can understand it, if these are the types of nanotubes that we think we're feeding to cell culture, but these are the ones that actually get fed, that there's some kind of a disconnect between concept and final result. And here's then titanium oxides exposed to bronchial alveolar fluid in the lung, showing that individual particles, once they're exposed to bronchial alveolar lavage, all of a sudden just group into these huge aggregates. And so the presentation of titanium oxide to the lung is not really at the nanoparticulate form, it's in these huge multi-particulate aggregates. Okay, so this aggregation then affects their toxicity. And there's many, many anecdotes to show how, if you don't pay attention to how you feed these particles to the cells, and you don't think about the relevance of how that would work in vivo, that you've got a huge disconnect. All right, observation number three, different labs get different answers. This is a simple slide showing that they sent the exact same well-characterized nanomaterials to labs A, B, and C in 3 different continents, North America, Asia, and Europe, and they got 3 totally different answers in terms of the IC50s and the IC75s for the same materials fed to the same cells under the same conditions. So there's some kind of problem with inter-lab reliability in terms of how we do nanoparticle assays, and this is being worked out now with a standardization routine driven by the Europeans that want to say everybody's got to do the same thing at the same time. But I can tell you it's like asking a millipede to jump, right? He can't get all of his legs off the ground at the same time, right? So we got that issue going. Observation number 4, there's increasing evidence that the classic cytotoxicity assays may not really be relevant for nanomaterials toxicity testing. And I've got all kinds of lists for why these cell culture assays do not approximate the preclinical animal models and why then they fail in terms of their predictive capacity for understanding some nanomaterials toxicity states. So I'm going to skip that then. So what's expected then in these in vitro and vivo correlations? Well, the cell lines, as I've shown you, has a reductionist approach that has certain limitations. And the observed trends that we see in cell lines are really not mechanisms. They're toxic responses for that particular cell under those particular conditions, which may or may not translate. So it really doesn't define risk or harm in these in vitro assays. We also have a problem when we move in vivo because there's species-to-species translational issues. And so we really have a connection problem with the in vivo case. The other question that's often asked right now is, so my students work on this 24-hour cycle, like, you know, they report to work at the crack of noon, right? They put the cells in culture by 3 p.m., right? And then they come back the next day at the crack of noon. Oh, that's 24 hours. So it's a 24-hour assay, right? And so what relevance is that for in vivo work? Cell cultures at 24 hours is an arbitrary time point, very short time scale, does not then reproduce the chronic, repeated cyclic exposure of what you might be eating every day or what we might be feeding somebody in a pharmaceutical dosage form every month for a chemotherapy. So this biodistribution is likely not meaningful in a 24-hour assay in culture. So my bottom line is that the validation of these new models will be critical. So in terms of assessing this in vitro nanotoxicity, there's numerous examples where in vitro and in vivo doesn't work in assessing toxicity. Glass fibers, for example, that cause oxidative stress and all kinds of cellular problems in animal models, in vitro, but in animal models, they really show no ability to induce disease. Kaolin, a clay product, is cytotoxic in cell studies, but when you put it into workers, it shows very little ability to cause disease in human workers. Single-wall carbon nanotubes don't induce significant toxicity in vitro in cell culture, but they cause interstitial fibrosis when inhaled into mice in a few studies. So we've got then all of these things where we're really not sure where we're going with this. I'm going to skip this one. And so observation number 5, my last observation, is that human models are not equivalent to mouse models. And just to say, don't worry, I had the same thing and they cured me, the mouse is saying to the human. And there are many, many cartoons like this. I've got a whole slew of slides. I could spend an hour doing that if you wanted to. So in terms of the animal GI model relevance, remember in rodents, M cells in the intestine, M cells are 10 to 50% of the cellular lining, as opposed to humans where it's 1%, right? So humans, we have this very, very small amount of particle uptake, but in rodents we see a very high amount. So when you see rodent uptake, I always think, oh, that's going to be highly exaggerated over what the humans would have. M cell markers to denote what the cells look like in the intestine are very species specific. So sometimes we don't even know if we can find the M cells in various species because we don't track the markers very well. And the rodent gut physiology, of course, was mentioned this morning, is very different. Here's the pH values of human GI fluids. Here's the pH values of rat GI fluids and the fed and fasted states. They're very, very different. So the oral tract is different in our preclinical models than it is in our in vivo target for humans. So in summary then, our in vitro methods, the nanotoxicities, I have to ask, what are the relevance of the current nanotoxicity studies? We're using transformed cell lines that are mortalized. We use low serum containing media, and that does not equate then to human host response with full serum with 4,000 proteins surrounding every cell. Particles, they aggregate in biological fluids, so we don't know what the physical state and the stability is in serum. You oftentimes are using supraphysiological acute exposures. In cell cultures, we dose with micrograms per petri dish. And to make that equivalent to a human dose is some fantastic kilogram exposure. So we don't really have a translation of the actual human exposure. And I present then some example here I don't have time to talk about. We have highly conflicting cell-based data to date. So you can go to the literature and you can find any answer you want right now for nanomaterials. Titanium's toxic, titanium's not toxic. Carbon nanotubes are toxic, carbon nanotubes are not toxic. And you can go find out with everything you want. So it's really confounding. I feel sorry for people who are not embedded in, I feel sorry for myself actually, but I feel sorry for people who are not embedded in this expertise because it's very difficult to tease out in the literature what you need as a validation for how you should proceed. So the bottom line. So what's the bottom line? It's right there. Okay, so the bottom line. So convincing evidence for harm from oral ingestion. Somebody missed that? I have to go back. It's right there. Okay, so for harm is oral ingestion. And so the convincing evidence in humans is either inadvertent or it's for deliberate or inadvertent exposure is scant. That is, we've tried to pump rats full of lots of stuff. A lot of them don't get sick with anything. We've also found anecdotes with humans where we don't find any harm to date. So the one example I could find actually come from Andrew Maynard's blog. 2009, the UK House of Lords received no evidence of instances where ingested nanomaterials have harmed human health. But still, large gaps in our understanding of how nanomaterials behave or how they affect the biology of living organisms, especially human health, further research is needed to enable effective risk assessment of nanotechnologies used in the food sector. So that's a quote from their report. And they then go on to list all of the specific points that need to be addressed in order to provide a toolkit to fill these gaps in the understanding of how nanotechnologies used in food. So in conclusion then, particle uptake has been shown repeatedly in animals. It's slow, it's less than 10% of the dose. In humans, it's less than 1% of the dose. Successful prediction of oral gastric particle uptake requires the process to be both predictable and reproducible, but yet it's not been so in humans, it's not been so in rodents, and it's not been so in cell culture. The limited extent of kinetics particle uptake, that is, it's very slow, make it unlikely that the process presents a risk for typical engineered nanomaterials approved for use right now. Unless we make some magic particle that can dance through the cell membrane and get into the bloodstream, this probably won't change. I can tell you that I have pharmaceutical colleagues who are looking to make that particle, then the situation will change. Specific GI targeting using biological ligands is expensive. It's an academic exercise right now. In my opinion, it's unlikely to go viral. It's unlikely to go into the food sector. It's just too expensive. Those particles that do enter the systemic circulation suffer from the first pass effect. That is, they finally escape the intestine, but they get caught by the liver. So in fact, the oral dosing that does get into circulation undergoes a whole other processing step by the liver and the spleen, subject of another 2-hour lecture that I give to my students, that in fact is called the first pass effect. So many of these particles are captured by this first pass system. I don't have time to talk about that. In vitro assays predict the in vivo uptake, very, very few, woefully lacking to date. And food and nutritional exploitation of nanotechnology will likely then exploit solubilization capabilities, but suffer from the same solid processing fate that current nanotech does in pharma. So I'm going to say we have to be cautious about how we choose our future. Somebody mentioned this morning, the garbage in, garbage out problem. To understand risk, we really need to accurately understand the dosing, the nanoparticle physical and chemical presentation to the biology. We understand the host reactivity, like the gut, and how the gut processes. We need to develop predictive and reliable toolkit to move from in vitro to in vivo. So with that, I'm finished. I appreciate very much being here. Thank you very much for the invitation.